Hello everyone, in this seminar we are talking about Poineers in Bioinformatics, to be specific, John KENDREW and Max Perutz. The presenter is me, Al-Huda, a master student of Bioinformatics in College of Artificial Intelligence, University of Baghdad, and it's supervised by Assistant Professor Dr. Rashad Khalidi. The seminar outlines are the history of bioinformatics, John and Max's work, and modern tools related to their work, with a case study comparing how the workflow was before and after their discovery. So the discovery of bioinformatics was a big step in the biology world because recently it became an essential toolkit for biologists. And by that I mean computers and specialized software. For example, if we have a DNA or protein sequence analysis project, we need to parse meaningful information in massive gigabyte-sized biological datasets. Virtually all modern research projects in biology require, to some extent, the use of computers. However, the very beginning of bioinformatics occurred more than 50 years ago, with desktop computers were just a hypothesis, and DNA could not be yet sequenced. But it was a thought. Here is one of the first automated peptide sequencers designed by William J. Dreyer in the late 50s. And it was a big leap, yet it was slow, it was expensive, and kind of impractical if you look at it. John and Max were the first people or scientists who determined the structure of a protein. Before that, they thought the protein had no structure, or they thought that the structure of the protein has nothing to do with its functions. So John and Max were the British scientists who jointly won the Nobel Prize in 1962 in chemistry for determining the structure of a globular protein, specifically myoglobin and hemoglobin. Myoglobin was for KENDREW and hemoglobin was for Max Perutz. They did this using an X-ray crystallography in 1957. This was the first X-ray photograph of myoglobin crystal in the late 50s, photographed by John KENDREW. Sausage model of myoglobin was the first published, let's say, semi-3D model that KENDREW made or presented, and it was hard to hide his disappointment when he first published it. He said he was surprised and with the unexpected twists of the protein. He was a bit shy since his model was, as he described, ugly comparing to the DNA of Watson and Crick. So one of the fellow crystallographers said, "It's a horrible object, but a beautiful work." A fellow crystallographer saw it and helped to print the myoglobin model as the first print appeared with the help of KENDREW. So this is the same model structure, or the sausage model, but in a print. You might think, how is that actually related to the modern tool? So KENDREW and Perutz were the first who determined the 3D dimensional atom structure of a protein using X-ray crystallography. This proved that proteins had a specific folded atomic level structure that determined its function. The breakdown created a new field, crucially generated a new type of data, which is the 3D protein coordinates. The need to share this data led to the creation of a protein data bank known as PDB in 1971. Which also led to creating the AlphaFold, or the AlphaFold 2 to be specific. It's an AI system from DeepMind that was trained to predict a protein's 3D structure only from its amino acid sequence. The entire training set of AlphaFold was the PDB, or the data set filled with the structure determined by the method pioneered by KENDREW and Perutz. As you can see, this is the PDB website. It has approximate 180,000 experimental structure determined by the technique pioneered by KENDREW and Perutz, served the ground truth training data for the AlphaFold protein structure database model. It presented in the same language and format as the experimental structure from the PDB. It can be manipulated, analyzed, and used for drug discovery in exactly the same way. It now includes AI prediction, stands on the shoulder of the first pioneers who showed us what a protein really looks like. As you can see, this is the real example of the PDB website. You can choose any protein and it shows its sequence, its structure, its information, and everything about it. And this is the real structure of myoglobin. In AlphaFold, you can see its information, its sequence length, and active sites and details you might need in your research. Let's assume I have sequenced a gene for a protein involved in a disease. What does the protein look like and how does it work? Without a 3D structure, understanding its mechanism is very difficult. So before the reality that KENDREW and Perutz gave us, we would have literally no idea what we would do. What is it actually? We had no idea that the protein even had a structure. But with KENDREW and Perutz's solution in the late 50s, the beginning of the 60s, the use of experimental methods like X-ray crystallography to determine the structure. It's still a slow, expensive, and technical challenging, often taking years. This is in the past, because with the modern workflow, we have the alpha fold revolution. First, we would identify a protein. Let's assume a researcher studies a newly discovered human protein linked to cancer. Its sequence is known, but we have no experimental structure exist. So we access the tools. Which tools? The researcher goes to the AlphaFold protein structure database. And then he retrieve the prediction by searching for their protein, and then gain insights. Because the researcher now visualizes the 3D model, they can see a deep pocket that looks like an active site or a region that might bind to another protein. This generates immediate testable hypotheses about functions and drug targeting. AlphaFold 2 could not exist without PDB that has approximates a 180,000 experimental structure, as we said. So how the AI was involved in AlphaFold 2? Basically, the AI or the model learned the rules of protein folding by analyzing the relationship between sequence and structure in this vast PDB library. KENDREW and Perutz provided the first examples, and the structure biology community, they inspired, built the textbook that AlphaFold 2 learned from. I can say that we are living in the direct legacy of KENDREW and Perutz. Because of them, we lived the creation of the protein data bank. People knew the need to share their protein coordinate data, which established the PDB, and this repository became the central library of 3D structural information, which also led to the training ground of AI, because the hundreds of thousands of experimental structure in the PDB, determined using methods pioneered by KENDREW and Perutz, provided the essential ground truth dataset that was used to train the revolutionary AI systems like AlphaFold. Also led the launch of a new science or field called structural bioinformatics, because this work created the entire field of structural biology, which inherently requires computational tools for visualization, analysis, and comparison of a 3D structure. The data generated became the essential ingredient that decades later enables computers to learn the rules of a protein folding themselves. Their experimental breakthrough is the direct ancestor of modern computational structure prediction. So we can say that these two pioneers did not just solve two protein structures, they provided the first key to understanding the molecular machinery in life. These are the references if anyone wants to check or to see the 3D structures themselves, and thank you for listening.